Well, guys, I'm really very excited today because I am reconnecting with a colleague, a friend, and someone who I have admired his work for a long time. I'm going to call him like a social media sensation, Dr. Bishal Gyawali. He'll introduce himself to you in a little bit. We are taping this episode at the end of April 2021. It's going to air in a couple of weeks. But Bishal is going to introduce himself, and I think there's some huge announcements that will be only heard for the first time ever on Healthcare Unfiltered. This is where we break the news, Bishal, right?
Yeah, thank you, Sadi.
All right, so Bishal, introduce yourself to folks, and let's break the news to everyone.
Thank you, Sadi. It's always a pleasure talking to you, and I miss seeing you in person. I miss meeting with you in Chicago, going out, hanging out for drinks, and so yeah. But thank you. My name is Bishal Gyawali. And the big news Sadi is referring to is I'm going to start as an associate professor in the departments of oncology and public health sciences in Queen's University, Canada, from June 2021.
So you got promoted within 2 years to an associate professor. It couldn't have happened to a better person. Many congratulations to you, Bishal. You work very hard and you work from your heart, and you deserve all the best.
Thank you very much,
just for folks who probably don't know you a little bit, just tell us a little bit about, we're going to talk about your recent research and your recent publication, because I thought it was just fascinating. I loved it. But just a little about you, where you went to school, background a little bit, and how did you end up in Canada?
Yeah, that's an interesting story. I refer to that to my journey as from Nepal to Nippon to the north. So I'm from Nepal. I did my, I was born there, grew up there. I did my medical school in Nepal, Tribhuvan University. And after graduating my medical school, I went to Japan for my medical oncology training. I did my medical oncology training there for 5 years. I also got a PhD there from Nagoya University in 2017. And I returned back to Nepal. I worked in a government hospital called Civil Service Hospital in Kathmandu for around 6 months as a medical oncologist. After which I moved to Boston to do a research fellowship in cancer policy with Dr. Aaron Kesselheim in the Portal Program on Regulation, Therapeutics, and Law group at Harvard. And in 2019 March, I moved from there to Queen's University in Kingston, Canada. And until now, I was working as a clinical fellow in oncology and an assistant professor in the Department of Public Health Sciences. And from June of this year, I'll be transitioning to an associate professor in both these departments.
Well, congratulations. So how many languages do you speak, buddy?
4: Nepali, Hindi, English, and Japanese.
Okay, so what we're gonna do is we have to say a few words in each language, so we see my goal. I need to get more listeners from Japan and Nepal. I have to ask you, Bishal, how's your family and friends and colleagues back in India and Nepal? I mean, we hear so much of these tragic stories, and I'm going to air an episode on that. I've already did, and I'm going to air another one, but I hope, what's going on? How are things?
Yeah, it is very scary, to be honest. And it is partly our own fault because we did not take the first 2 waves seriously. And, I don't know exactly how to explain this, but when the whole world was being hit by COVID, it did not affect Nepal, India as bad as it did in these other parts of the world. So in a sense, when I was talking with my family and friends in Nepal, for the first couple of weeks, they were scared and they were following all the precautions. But slowly they realized that probably they are immune from COVID. Like, people were not dying, and there was not much hospitalization. So people started opening up, and that opening up became so intense that people forgot that the rest of the world is still suffering from COVID. So I was surprised that people were having wedding parties, political rallies, as if nothing had happened, and nothing was happening until this Yeah, what we call third wave here, or probably just a second wave back home. But when this new variant started, this double mutant variant, now this is having very severe consequences, and like, I guess what was happening in New York and Italy and those uh, big hits during early pandemic is exactly what's happening now in Nepal and India. But the problem is we are not equipped to deal with these huge outbreaks. Our medical system has always been strained even before the pandemic. And people are not following the guidelines well, because as I mentioned, the first wave did not hit us that bad. So we had a false sense of confidence and complacency that probably our part of the world is immune to COVID virus. So
But
people,
Vishal,
yeah.
do you remember there were a lot of thought leaders and
Mhm.
experts that were trying to that they did suggest there may be some plausible scientific biological reasons why folks and people in that region of the world were not as heavily predisposed to COVID, uh, whether it's BCG, whether it's you remember those?
Yeah. I do. There are people were trying to explain that differences, and I don't know if any of those hypotheses are true, but I think our main mistake was that we did not think that there could be mutations and the newer variants that could affect us, and we did not use. Yes, we suffered less compared to the rest of the country. That's true. We did not have as much effect of the disease during the first wave, but we did not use that time to prepare ourselves better for whatever wave it would eventually come. We did not, instead of focusing our resources on strengthening the health systems, we instead had these big political rallies and parties, and we did not we were lucky in that we got plenty of time, uh, but we were unfortunate in that our system did not use that time to prepare ourselves better.
Yeah, well, I hope things really just get better, better fast. I
Yeah,
really
it's especially scary for people like us who are away from the family, and
absolutely.
yeah, all I can do is just talk to my parents like I'm talking with you now in a video call, but that does not compensate for the fact
Oh,
that
it's
yeah,
not the same. It's not the same.
yeah,
And I hope you get
yeah,
to see them very, very, very soon.
yeah.
Well,
Thank you, sir.
let's distract you a little bit. Let's distract you a little bit. It's a good distraction. I've asked you to come on the show because I mean you're one of the most prolific researchers and writers that I follow, and I follow your work. And one of the things that you are passionate about is clinical trial design, and you're appropriately critical when trial is not adequately designed or powered and all of these things. And even, I mean, we're taping today several or a day or two after some recent ODAC decision that you were vehemently opposed to, and so forth. But I want to talk about optimal clinical trial design
Mhm,
with you, because you wrote about this and you
mhm,
identified certain aspects of clinical trial design that you believe should be fulfilled. So
mhm,
that's really the frame of our conversation. So
mhm.
let's level set first of all, Bishal. What led you to write that paper about clinical trial design? I mean, why did you even write it?
Yeah, you caught me at a little sad time after the ODAC meetings. I'm a little depressed that our community will change or the system will change for the better. I'm a little depressed right now, but when I started writing that paper, I was very much encouraged, and I thought naively that what people need is education, and if they are educated, probably they'll change. But now I'm having thoughts that maybe probably people already know, but they are just choosing to turn a blind eye, and that's a big difference between not knowing versus knowing but turning a blind eye. So when this all started,
I love that, but
actually,
I love that, by the way. Not knowing versus knowing and
yeah,
turning a blind eye.
yeah. So this all started last year at the ESMO annual meeting. And I have to give huge credit to the chief of the ESMO MCBS. This is the magnitude of clinical benefit scale, ESMO MCBS working group, Professor Nathan Czerny from Israel. So first of all, he invited me to be a member of the extended working group for the ESMO MCBS committee. And while working through various meetings in that committee and thinking more about how we can grade the magnitude of clinical benefit for different cancer drugs appropriately, Nathan asked me if I would do a presentation at the ESMO annual meeting about different issues with trial design that can give a falsely high score for a cancer drug. So the readers may or may not know, the audience may or may not know, there is something called the ESMO MCBS scale. So this is the scale that grades cancer drugs from 1 to 5 in the advanced setting and from A to C in the adjuvant or neoadjuvant setting. And higher the score, better is supposed to be the clinical benefit of the drug. So a drug with a clinical benefit score of 4 or 5 is considered to have substantial clinical benefit, versus a drug that has a score of 1 or 2, which is supposed to have very minimal clinical benefit. And this scale is now adopted by the WHO in discussing their essential medicine list and various countries in Europe and elsewhere to decide which drugs to reimburse versus which drugs not to reimburse, and those policy decisions. And this score, now I'm just making a generalized statement, there are nuances to it, but this score basically takes into account the difference in median survival times, the hazard ratio, confidence interval, toxicities, quality of life, and so on to come up with that value. So during that annual meeting talk, in my talk, I made a point that yes, that tool is quite important because it gives us an idea about prioritization of drugs. But what about certain flaws in trial design which cannot be captured by that value? Like if you are for example, if you are using an inferior control arm, then your median survival times will look great, the hazard ratio will look great, everything will look great, but there is the issue that the trial did not use an appropriate control arm, and that's why everything is looking better. So those issues, how are we going to control for those issues, was my question.
Right. So your role was to identify the issues that could artificially improve the score of a cancer drug.
Yeah, and make a drug look better than it actually is. So I presented at last year's ESMO virtual annual meeting about these certain trial design issues that could inflate the scores and make a drug look better. And and it shows like the reason I said I told you that I was encouraged is because after that presentation, the MCBS team, the ESMO team as a whole, took those points so well. They thought like, yes, this is really an issue. Like they could have taken it in a wrong way, and
Sure.
people can always defend and say that and defend the trial designs. But the whole team took it so positively, and Nathan encouraged me that like many people will not hear the talk, but if we write about it, then many people will read the paper, and it can be used as a source of reading material for trainees as well. So Nathan and Elizabeth DeVries, both chairs and coach and past chair of the ESMO MCBS team, they encouraged me to write it, and then we started writing about it. And once you start writing about it, things get expanded, and from 2 examples, 4 to 6, and then
So
we try to be as
how many points did you identify that could cause inflation of a drug score?
So we identified 10 points in total.
Okay, well let's go over each one, and I'll try to play a counterpoint for each one, so the audience will listen to enjoy a little bit. And then, okay, first point.
So we divide these 10 points into 3 categories: one as design issues, and the other as analysis issues, and other as implementation issues. So first, with regards to design issues,
I can tell by the way that your daughter probably is screaming in the background.
Yeah,
It means
she's
it
playing.
means you're at home. You're not at work.
Yeah, that's why you have this virtual background, because
Yeah.
I can't show you my real background, what it looks like. Unlike you're very tidy
The
and
nice thing of
graceful.
Zoom. The nice thing of Zoom.
Yeah.
All right, let's go over the first point.
Yeah, so the first one is the substandard control arm issue. As you know, this has been a real issue in recent years. Take the example of the Polo trial in pancreatic cancer. You can take the example of the PARP inhibitor trials in prostate cancers. Um I know that Binay has already ripped these trials apart. Uh, mm,
But Bishal, whenever we talk about the substandard control arm, and I do totally agree with you that some of these are definitely substandard, but oftentimes the pushback that we get about the substandard control arm is twofold. One is that it may be a substandard control arm in the US, but in low middle income countries, and I know you're very passionate about that, this may be an acceptable control arm. And in a global trial, we need to allow this because that's really what folks in these low middle income countries could get. And the second pushback we usually get is we started the trial in 2014. This was an acceptable control arm. The trial read in 2020, and during that time, the control arm changed. It became very difficult to make these changes from a regulatory perspective on that trial. How do we respond to these two pushback? And do you agree with
no,
them?
those are excellent points, especially with regards to the first point that the actual standard control was not available in rest of the world. So when we are running a trial globally, it was not possible to offer the best possible control arm to the participants outside of US or Europe. But I think there are certain problems with that argument. Let us talk, taking some real examples. Let's name names. There is this new Empower trial of a new anti-PD-L1 antibody, semiplimab, in lung cancer. So it got FDA approved. So this trial ran in rest of the world, semiplimab versus chemotherapy. And after we already knew that we had all those positive trials of pembrolizumab in lung cancer, and for patients who are PD-L1 positive, we ran a trial outside of US, randomizing patients to receive either pembrolizumab or chemotherapy. So for me, that is clearly an inferior control, and that has two consequences. One is that if that trial was not used as a basis for approval in the US, then that would have been a different story. But that same trial is being used as a basis for approval in the US. And the second, more importantly, about global health, this is a recurring theme in global health research, and especially in global cancer trials. So I want to stress this point. People argue that, okay, like at least the participants in the trial got access to a PD-1 inhibitor, because these patients would never have access to PD-1 inhibitor if they were not a part of the trial, is what people argue, right? Because let's say we are running this trial in Nepal. I'm just giving an example. We did not run this trial in Nepal, but let's say we are running this trial in Nepal, and my Nepalese patients would never have access to a PD-1 inhibitor. But now the argument is that we should feel happy because at least some of my patients got access to PD-1 inhibitor because they were a part of the trial. How big is the trial? A trial involves like 500 patients, right? And of those 500 patients, half of them will be randomized to control arm. So it's 250 patients. Now, what is the lung cancer population of that country, right?
Yeah.
And the lung cancer that, like in a global trial, maybe there are 2 patients, 3 patients, 5 patients from one particular country, or even if the whole rest of the world, if we consider as one bucket, maybe that is like 100 or 150 or 200 patients who got access to a new drug. So the question is, after that trial was completed and we knew that this new drug semiplimab is better than chemotherapy, or how many of those patients, A, will continue to have access to the drug, and B, because these patients participated in a trial for the ulterior good of future patients, how many of the future patients in that country will have access to the same drug? None. So
Is that
because
true? So I didn't know that. So basically, that checkpoint inhibitor that was used in that trial is not going to be made available in that country where the trial enrolled like 250 patients?
maybe they will continue for those particular patients who were enrolled in the trial, but not for the other patients with the exact same scenario, right? So because it is out of affordability for those countries and for the patients. So this is a classic example of, I may be being harsh here, but I am emotionally involved as well, being a global oncology researcher coming from what the world calls the rest of the world. I think this is research parasitism, what people call research parachute. I think this is this is an example of research parasitism. You are, in a sense, you are just using these patients so that you can get your drug approved in the US, because no patients in the US would sign up for such a trial. So in case of collaborative global research, there should be some benefit for the global community. And what is the benefit here? Should we just be happy that, okay, 2 of my patients got access to control?
That's a very good point. I see your point. I think I think the argument for that is rather thin. It's not really very strong. I see your point. How about when the control arm changes, like, while you're running the trial, something discovered?
Now that is a more valid argument. I agree, and this has happened a number of times. To be honest, the ideal thing is, and the ethical thing is, if the control arm changes during the running of a trial, then there should be, like, there are so many amendments to a trial protocol. So I think it is ethical and imperative to let the patients know that the control arm, like the standard of control, has changed. And ideally, if you are not a part of this trial, you would have received this control arm, which is a better control arm that we already know. So that would be the ideal and ethical thing to do. But I would consider that this argument is more valid than the previous argument. And sometimes it may not be logistically feasible, but it also depends on how, it also depends on the trial duration and the years of the trial. So if a trial completed in 2015, but the standard of control changed in 2014, that's very much understandable. But standard of control changed in 2014, but the trial is running until 2020, then that is not justified.
And Bishal, there are a lot of people that would say, but deciding on a control arm sometimes is not easy when you're running a clinical trial. I mean, I've been part of cooperative groups when I was at the university, and I would attend the cooperative group meetings where there's a lot of discussion into the design of this particular trial, and there's so many many discussions into what's the control arm for this trial, what's the experimental arm, and I could tell you it's not easy, honestly, to reach a consensus. So many clinical trialists will push back and they will say, well, it's not really easy, and sometimes we have to figure out the best of worst evils, or whatever it is that we say.
I would put a counter argument, like with these trials, why would you always, like with all those debates going on, why do you always see that the conclusion is always towards using an inferior control arm? Then why not use something better than
because you need the trial to be funded, because otherwise the trial won't be funded.
Okay, for example, let's talk about Olympiad trial. This is the trial of olaparib versus physician's choice chemotherapy in triple negative breast cancer. So the funny thing is, I guess because of those debates about what should be the control arm and nobody reached to a consensus, they said, let's use physician's choice as the control arm, right? But when they say physician's choice control arm, why did they specifically exclude platinum agent? They say physician's choice control arm, but you can choose among these 3 different chemotherapy, but you cannot choose platinum. So, like, these are the things that make it clear that there was some motivation to play with the control arm so that it does not look as unethical, that it would not be approved by the IRB or the regulators, but you could still have that advantage of
No, very,
yeah.
very, very good points. Okay, what's the number two in clinical trial design? We talk about the control arm. Number two.
And number two is about the use of surrogate endpoints.
Okay, let's talk about that.
Yeah, yeah. This has mm, mm, we have done a lot of work about this, and I think like whenever we talk about surrogate endpoints, first there are a couple of things we need to consider. A, what is the validity of the surrogate endpoint in that setting? Like all surrogate endpoints may not be same, right? Uh, and the FDA actually in I think 2018 they did a good work by publishing a table, a list of all the surrogate endpoints that they have used in the past or they are willing to consider in the future, not only for cancer but for all diseases. And in that table, if we look at the cancers, we see that the FDA has like put all tumors together and said like response rate is good enough for accelerated approval or regular approval. But I argue that you can't just say or PFS or DFS for that matter. PFS is a good enough surrogate for all solid tumors or a bad surrogate for all solid tumors. There might be some setting where it is a good surrogate, and there might be some specific setting where it is a bad surrogate. And if we are using a certain surrogate as a basis for regulatory approval, it is mandatory for us to also show the validation results. Like what is the correlation coefficient? What is the 95% confidence interval? Why is this surrogate a valid surrogate in this context? Or why it is not a valid? The surprising thing is, I think you may have read that paper in 2019. We published that accelerated approval review of FDA in JAMA Internal Medicine. And we saw that the FDA was, in some cases, using the exact same surrogate for giving accelerated approval as well as for giving regular approval. And that makes absolutely no sense to me. If you think that a surrogate is weak enough that it does not deserve regular approval and gets only accelerated approval, how can the same surrogate now be used and be said that it is good enough for regular approval?
So you're not against using surrogate endpoints. You are willing to use surrogate endpoints if there are studies that have demonstrated their validity in a particular context.
Yes.
Can you give listeners an example where you are in support of using a surrogate endpoint?
Yeah, like if there is a good validity, for example, I like off the top of my head, I think DFS is a valid enough surrogate in case of colorectal cancer.
Okay.
And similarly, I think DFS is also a good surrogate for HER2 positive breast cancer.
I see. Okay. But this situation.
Yeah, but we can't lump together. Like we can't say for all breast cancers. We need to study that for specific situations. And even in lung cancer, DFS is a good surrogate for OS when we're using chemotherapy or radiation, but we don't know whether it will hold true for targeted therapies and immunotherapies.
I think the issue with surrogate endpoints, many people will contend that because we're making good progress in cancer, which is good, right? I mean, we're all happy whenever we make progress in cancer. Overall survival is extremely difficult to be the only endpoint. It's going to delay drug approvals, patient access, and things like that if you put overall survival as a blanket statement for every drug approval.
I had a good answer for that until a week ago, but this ODAC meeting ruined everything. This ODAC meeting ruined everything because US
Tell us about that, by the way. Tell us about that.
Because US had the best system. The US had the system of accelerated approval on the basis of surrogate endpoints. So it was the best of both worlds. Like there are two groups of patients. One group of patients, you might have seen this in a clinical practice, that there are certain patients who would even refuse to get even like curative treatments because they just they have very high threshold for any receiving any therapy. They are afraid of the side effects. Even like I had to the other day, I had to convince a patient with metastatic colorectal cancer to get first line FOLFOX, like which is the standard of care. Did not want any chemotherapy at all. And then there are other groups of patients who want even all investigational therapies, like whatever is even in just being discovered in the bench, they are willing to take that chance. So there are two groups of patients. One group of patient needs to have certainty of benefit. I need to be confidently able to tell that patient that yes, if you take this drug, you are going to live longer and better. Hopefully both, if not both, at least one of them, longer or better. And then there are other group of patients who says, no, I need early access to therapy. I'm willing to take all the chances. Now, the US system is a very good system, the accelerated approval pathway, because it gives a balance to both these patients. It gives early approval on the basis of surrogate endpoints. So the patients who need early access to drugs will have their wish fulfilled. But it also mandates that, but you guys should conduct a confirmatory trial. And only if the confirmatory trial demonstrates clinical benefit, you guys can stay on the market. And so after that happens, the other group of patients are also happy because we can tell them, yes, now it has been confirmed. This drug does improve clinical benefit. So you should take this drug. So this is such a perfect system. But last week, what we saw is this accelerated approval is a package with two parts of the equation. But what we saw is the whole system is favoring only the first part of the equation and it's just ignoring the second half of the equation. Like only giving early access, approving early part of the equation is being honored. But the last half of the equation, that if the clinical benefit is not confirmed, the drug approval will be withdrawn or revoked. That part of the equation is not being honored. So this is in a sense, this is dishonesty. We are not honoring the contract. To be honest, Adi, I was surprised when the ODAC meeting happened. Because at first, my first thought was, why do you even need an ODAC meeting? The accelerated approval law is clear that if the confirmatory trial fails, the approval should be revoked or withdrawn. So the FDA has the
why? But let's talk. Why did the ODAC then voted yes? Like why? Why do you think that is? I mean, they were, I don't know how all of the members of the ODAC. I mean, it was unanimous. What are we?
Yeah. So my big surprise, as I was telling you, is first I was thinking we did not even need an ODAC meeting. The FDA could simply revoke the approval. And in fact, four approvals were withdrawn even before the ODAC meeting, right? The voluntarily withdrawn by the industry. I think they are regretting now that if they had not voluntarily withdrawn, maybe they would have stayed on the market. And the second thing was I was giving benefit of doubt to the FDA. And I was thinking like, okay, the FDA is trying to take good steps. This is good, at least like they will have an ODAC meeting and then they will withdraw the approval. But as you mentioned, in 4 out of 6 of those discussions, the ODAC committee, in fact, voted to maintain the approval. And in some of those cases, it was unanimous. It makes no sense, Sadil. Like, let's look at hepatocellular cancer, for example. There were 2 drugs being discussed, pembrolizumab and nivolumab. Both of them are same category of drugs, similarly bad. Like both of them failed to improve outcomes in the confirmatory trial. And pembrolizumab gets 8-0 unanimous vote to be maintained, and nivolumab gets a majority vote not to be maintained. This makes absolutely no sense.
Why? Like,
I was speaking with a journalist.
I want to do a podcast on that. I have to think maybe somebody from ODAC will actually be willing to come on the show. I'm not sure they will, but anyway. But that's a good point about surrogate endpoints. Let's move to point 3. We've got 10 points.
Okay. The third point is about crossover. So
okay, let's talk about that.
Yeah. So if there is a drug already approved in first line, if there is a drug already approved in second line, and you are trying to test whether that same drug is effective in first line, then I think there should be mandatory crossover design built into the trial, because that drug is already approved in second line, right? So the control arm patients, when they progress, they have to get access to that drug. Otherwise, it is clearly unethical. Or even in the case of adjuvant and relapse setting, this osimertinib ADAURA example, we know that osimertinib is effective as first-line metastatic treatment for EGFR-positive lung cancer. Now we want to test whether it is effective in adjuvant setting. Then it is mandatory that the control arm patients, when they relapse, they should get osimertinib. So this is appropriate crossover, mandatory crossover that needs to happen, but does not happen. So that is one problem with crossover. The other is the other way around. When crossover should not happen, but it still happens. For example, this Cipulation T in prostate cancer, it was never approved before. Nobody knew whether it was effective or not. So they tested it in first line. And when the control arm patients relapsed or progressed, they were crossover to receive this vaccine. So in effect, what happened was these control arm patients had delayed access to docetaxel, the treatment that actually works, because they were crossover to receive this new experimental drug.
I see.
Yeah. So in fact, if you look at the results, the response is just, there is no difference in response. It's just one patient versus 0 patient, I think. Like there is absolutely no response. And if you look at time to tumor progression, there is absolutely no difference. But if you look at OS, there is a difference. Now, how does that happen? Because the drug is ineffective, but these control arm patients did not get a better drug in time. So the control arm did worse than what would have actually happened. Yeah. So there are these two issues with crossover. One should have happened, but did not happen. The other is should not have happened, but happened.
That's really, yeah, you're right. So we talked control arm, surrogate endpoints, crossover. These are really very important. Let's go to point number 4.
The point number 4 is early stopping of clinical trials. Now, this is, there are two main issues with that. One is, I don't think it's even ethical to stop a clinical trial early based on surrogate endpoint. Like if there is a clear difference in survival that exceeds the threshold for stopping, then okay. But we are seeing more and more trials being stopped early based on those differences in surrogate endpoints, which is a problem. And the other thing is,
can you give us an example, Bishal, of something like that?
Like even ADAURA was stopped on the basis of DFS benefit. Like OS benefit has not been confirmed yet. It was stopped early. The trial was terminated early based on DFS benefit alone.
It was, and I know you're a huge fan of ADAURA. You wrote how much you love the ADAURA trial. I'm being sarcastic, of course, but people would argue, I mean, if the trial's primary endpoint was disease-free survival and it was met, that's why it was stopped. Like, you know, people will say, we're not stopping early. We met the primary endpoint, so we stopped.
No, it was stopped early because it crossed the early stopping threshold. But yeah, that's my problem. When we are designing a trial and we are putting in that feature of the ability to stop the trial early, the whole purpose of stopping the trial early is, the philosophy is that the control arm patients should not be at a disadvantage, because if we know early that the drug works, then these control arm patients should have access to the drug. So we are stopping early to protect those few control arm patients from having access to the drug, and after the trial is complete, the rest of the patients will also have that information. But in case of ADAURA, as you can clearly see, there is still debate, right? We are still debating the consequences of ADAURA and whether all patients should get this drug or not. So in a scenario where even after publication of the trial, even 6-8 months post completion of trial, publication of trial, we are still having that debate about whether ADAURA is really practice changing or not. How is it unethical to continue the trial? Or how is it ethical to think that the control arm patients will be harmed because we did not stop the trial early? And by stopping the trial early, we are deprived of having properly powered continuation of the trial for the hard outcomes. So I'm not against stopping trial early. I'm against stopping trial early on the basis of surrogate endpoints only. If there is a clear signal of difference in survival, then of course it is unethical to continue the trial and we have to stop it. A, and B, now this is not an issue with the design, but this is an issue with the interpretation. One thing that we need to keep in mind is whenever a trial is stopped early, it is stopped early because it crossed that threshold of stopping boundary. So whatever hazard ratio and confidence interval we see, it will look better than what it would have been had the trial completed in normal time. So we might see like an impressive hazard ratio, and we might jump, wow, this is an impressive hazard ratio. But that is so
But
because the trial was started early.
why can't we stop based on a surrogate endpoint if this is the primary endpoint of the study? Like, in other words, take away just in general, let's say we're designing a trial, me and you, and the primary endpoint is a surrogate endpoint. It's PFS, it's whatever it is, and we reach that boundary that we met the primary endpoint. Why not stop?
No. If the primary endpoint is a surrogate endpoint, we can stop it when the endpoint is met. Normally, I'm not comfortable with putting in that provision of early stopping.
I see.
Based on, yeah.
I see. Got it. Okay. Point number 5.
Point number 5
I can't believe you found 10 points to critique clinical trials. My goodness. You're so difficult to please. Go ahead. Number 5.
Oh, I thought you would say there were only 10 points. So the other is about single arm trials, the response rates and duration of responses seen in single arm trials, because one previous study published in JCO, I forgot the exact year, showed that for cytotoxic agents, the response rates are inflated in single arm trials versus the same agent, same tumor in a randomized trial. And our recent paper in Journal of NCCN, we tried to study the same thing for targeted agents. And even in the case of targeted agents, we saw that especially the duration of response was quite inflated in single arm trials. So this is a caution against, because you see more and more drugs being approved, sometimes even regular approval based on phase 1 trial, phase 2 trial nowadays, because the response rate and the duration of response was substantial. So this is a caution that says the duration of response of, let's say, 8 months that you saw in a single arm trial is probably inflated. If you ran a larger randomized trial, then the real duration of response probably is somewhere around 6 months or 5 months.
Yeah. So would you not do single arm trials now, or would you do them as exploratory? Like
No, I would do a single arm trial to make that decision whether or not the drug is good enough to be tested in a randomized trial.
Okay, got it. So you would not approve a drug based on a single arm trial?
Yeah, I would not. But if at least until a week ago, I had a compromise that, okay, even if you had to approve, there is accelerated approval pathway, and later you will confirm clinical benefit and withdraw it. But now that has gone.
All right. Okay. Point number, are we at 6 now or 7?
I think we are at 6.
Okay.
The 6th is about the use of non-inferiority design.
Oh, that's a tough one.
I think we have to think of non-inferiority design in terms of 2 issues. One is whether it is justified or not, justified non-inferiority design versus non-justified non-inferiority design. And the second is the non-inferiority margin.
Okay. So what's the justified versus non-justified non-inferiority design? Just a couple of and I always think listeners would probably relate more if there's an example.
Yeah, like justified is if the newer drug has any advantage, then sometimes you can do a non-inferiority design because certain compromise in efficacy might be acceptable. So certain advantage in terms of, cost. The newer drug is cheaper than the older one, or which never happens. And the other one is in terms of ease of administration. The newer drug is oral. Standard of care was injection, so the patients would prefer oral. Third, less frequent. The standard of care is every week. This one is every 3 weeks,
right.
Or has a better side effect profile. So if any of those 4 criteria are met, then it's a justified non-inferiority design. You could justifiably argue that because it's an oral, patients are maybe willing to tolerate up to a 10% increase in the risk of recurrence. So we did a non-inferiority trial.
When you talk about the margin, is that arbitrary chosen, like the margin of non-inferiority?
That's the big problem, and that's what I have been shouting for a long time about. Like, who decides this margin? I think we should ask the patients. Like, all non-inferiority design trials should begin with a survey of the patients, saying up to what % increase in the risk of recurrence or increase in the risk of dying would you consider acceptable for this benefit. I'll give you one example. I was shocked to see this. I don't know whether I can and find that exact example and quote the exact number. Yeah, there was this trial in sarcoma about pazopanib versus doxorubicin as first-line treatment in metastatic sarcoma.
Mhm.
Now, non-inferiority design is justified, right? Because doxorubicin is toxic chemo. Pazopanib is also toxic, but at least it's oral, so probably non-inferiority design is justified.
Right.
But you know what the definition of non-inferiority is? It's a confidence interval hazard ratio of 1.8. 1.8. I had never seen anything in that like that in my life. I have seen like 1.3, and I was I was upset that, oh wow, people are thinking up to 33% increase in the risk of dying is non-inferior, but this was 1.8.
That's 80%.
Yes, I don't know why they didn't
Wow.
make it two, double the risk.
Okay. All right. That's
Yeah.
pretty interesting. So, but I like your idea about non-inferiority. I think, I'll confess, those are tough studies for me to interpret. Like personally, when I read a study that is doing non-inferiority, I always feel I need help with the stats portion or the methodology portion when there's non-inferiority. I kind of feel it requires really a good critical eye in statistics. Okay, point number 8.
Point number 8 is about post-progression therapies. So, until now we're talking about surrogate endpoint, but this is an issue when overall survival is an endpoint. If you have overall survival endpoint, but after disease progression, the patients did not have access to proper post-progression subsequent therapies, then that can make your drug look better than it actually is. Like, mm, you know,
Yeah, but Bishal, and remember, once you progress on study,
mhm,
right, you
mhm,
you pretty much the clinical trialist. Okay, let's say you are the PI, you're the clinical trialist, and I'm your
mhm,
patient. Once I
mhm,
progressed on study, the study
mhm,
cannot dictate to you
mhm,
what you are going to treat post progression. It's really what you decide. I mean, no trial dictates post progression what you need to treat. Clearly,
mhm.
it's a limitation because people could treat post progression differently, but that's not the fault of the funder of the clinical trial because it's all over the place in the real world, no?
That also ties in with our first discussion about this running global trials. So if you are intentionally running trials in such a way that most of your, like the control arm patients, like you're running in countries and you're confident that okay, they are not going to have any subsequent therapies. Uh,
Well,
uh,
I mean, I don't know what they're going to have. My point is, I have no control over what they have. I designed the trial, and then post progression, I follow these patients until death or whatever it is, but I have no control. You may end up sending the patient back to their community oncologist or the referring physician, and you still know what they got, but you cannot tell that physician you have to, you cannot have a homogeneous post progression therapy.
No, yeah,
Patients
I
never.
agree with that. You may not have homogeneous, like similar post-progression therapies in each arm, but what but there are two things to that. One is that at least it may not be the same treatment, but th- you would see that like similar percentage of the patients got any subsequent therapy,
Okay.
right? Because if only 10% of the patients in the control arm got any subsequent therapy, but 90% of the patients in the experimental arm got any subsequent therapy, then that confounds overall survival, and that's why we are seeing the overall survival
Yeah,
difference.
no question.
Like we have seen trials of endocrine therapy, like you know, uh, ribociclib in first line hormone receptor positive breast cancer patients, and we know that usually women with breast cancer who are hormone positive would at least have like 4 or 5 lines of treatment. There are so many endocrine therapy options, and then you also have the option of chemotherapy. But if you are seeing a first line endocrine receptor positive breast cancer trial, and you see that like 50% of the women did not even go to second line therapy, and all of them have received just
No,
that first line treatment.
I'm not disagreeing that it's a big limitation. There's no
Mhm,
question. I think
mhm,
I totally agree with you. It's a limitation. I think what I'm pushing
mhm,
back against is I
mhm,
don't think post-progression therapy is the fault of the funder or the manufacturer, because I don't know how you would design a trial where you have kind of balanced post-progression therapy and similar post-progression therapy. But certainly, if there's significant imbalance, it does confound the interpretation of the results. I just can't blame it on the pharma, I guess.
No, I'll partially agree with you on that. I take a point. My argument is, definitely it's more related to interpretation of the trial. So when you see such trial and you are interpreting the results, you need to keep that in mind, A. And B, at least when publishing the trials, yes, there is no control over what patient got which treatment, but we should, like, the readers should be supplied with the information about what percentage of the patients received subsequent therapies and what therapies they were.
So you really want some, I guess what you're trying to get at, and I agree with you, is in some reports and some clinical trials, we don't have transparency into what the post-progression therapy was, so we can properly interpret the results.
Yes, that's right.
Okay.
And I have issue with, if it is happening, I don't know whether it's happening or not, intentionally running the trials in some part of the world, knowing that the patients there will not have access to subsequent therapies, and so that will make a difference in the outcomes.
Point number 9. I believe we have 2 more. For listeners, Bishal was going to have 30 points, but then he decided to go to 10. Point number 9.
Sorry, sir. This is point number 8.
8. Sorry.
So that is about quality of life reporting, because this is more related to the ESMO MCPS tool, because we give 1 extra credit if quality of life is improved, and we give 1 penalty. We take 1 point if quality of life is degraded. But what we know is there is a lot of publication bias against publishing negative quality of life data. So if quality of life data is not published, then they don't get penalty, because we penalize if the quality of life is worse. But if the quality of life data is not being published for many years, then probably that means the quality of life was worse, and they intentionally did not publish it. So,
but what if some trials did not in the trial itself there was no they were not capturing, I guess, quality of life data in the trial, so there's nothing to report on?
I understand that for like old trials, but for new trials, I don't think there is any excuse not to capture quality of life, because that's such an important outcome. We need to know how the patients are feeling.
I agree. I just don't know if it's, have you noticed that they are mainly included in all contemporary trials right now? Maybe it might be worth taking a look, to be honest. This is our project. It's about a time since we write a paper together. Look at clinicaltrials.gov and see the percentage of trials that do incorporate quality of life metrics.
Yeah, I have some old data. In 2018, I published this in International Journal of Cancer. We looked at, we did similar to what you just suggested. Only 50% of the trials had quality of life data into the trial design, but 25% of them who had quality of life into their trial design did not report that.
Yeah, wow. So, maybe worth a refresher. Let's talk about that offline. Okay, number 9.
Number 9 is about subgroup analysis, and this is...
Oh, I love that. I love subgroup.
So, the whole issue about post hoc analysis and making conclusions based on post hoc analysis. And the other issue is not conducting subgroup analysis when it is not supposed to be done. And this is very important in the context of the first ODAC meeting for atezolizumab in triple negative breast cancer. I find it so surprising, Sadi. The argument was that, we had 2 trials. The first trial led to accelerated approval, and then there was a confirmatory trial. The first trial, 130, showed that there was no difference in OS, and the trial statistical plan says OS in PD-L1 positive subgroup will be tested only if OS in the entire population is positive. So the entire population is negative, but they still did the subgroup analysis, and voila, there was OS difference. And they said, wow, this drug improves OS, although we cannot report p-value because we are not supposed to do so. And that gives accelerated approval. And the accelerated approval says, do a confirmatory trial. They do a confirmatory trial 131, which, no surprise, is negative. OS did not improve. Even PFS did not improve. So what the correct conclusion will be? The correct conclusion will be the first OS difference that we saw is a false positive, because that violated our own statistical plan. We did a subgroup analysis that we were not supposed to do. So whatever we saw was a false positive. The confirmatory trial says that's not the case. So the approval should be withdrawn. But what happened is people argued that the first subgroup analysis was a positive. Probably this confirmatory trial was a false negative. This is basic stats, and I don't know how to even make an argument against that.
Don't worry, you're going to have a stroke one day. Take aspirin. Take aspirin. You need to meditate and take aspirin. Okay, number 10.
And the final point is about informative censoring. Now, to be honest, even like I myself was not much aware of this issue until I started to really dive into this. And I think this is a point that many oncologists are not aware of, including myself from 6 months ago. So this informative censoring is, ideally when patients drop out from the trial, we, for other reasons, we assume that that is balanced between the two arms, between the experimental and the control arm. So that is called non-informative censoring. But if more patients are dropping out from the experimental arm because of toxicities and being unable to even come to the hospital and things like that, that is called informative censoring. Because now what will happen is these patients that are really sick or that did really poor, they are excluded from the analysis. So now the experimental arm is going to look much better than the real one. And this is especially important when the primary endpoint is a surrogate, because overall survival, there is no confounding.
So, but if there's enough patients that drop in both arms, it's okay when you censor those.
If there are equal number of patients, roughly equal number of patients for similar reasons, and that's the basic assumption that we have.
So this issue is when there's an imbalance in the number of patients. I see. Okay.
Who drop out and for reasons that are actually associated with the treatment. And one good example is the Bolero-2 trial of everolimus in breast cancer, in hormone receptor positive breast cancer. Because here what we see is there is a big difference in PFS, but there is no difference in OS. And if you look at the toxicity rates, the toxicity rate is quite high with the drug. So this gives us an indication that many patients dropped out because of toxicity, and that did not reflect in the PFS, but ultimately the OS turned out to be the same.
Oh boy, Bishal, that is really well. First of all, thank you. This is really very good. I loved the paper myself. I like that we are analyzing each point alone. I know that you mean well. You wanted to educate a lot of people about clinical trial design. Do you, I don't know. Where do you think this is heading? What do you think there's any opportunity for clinical trial reform? Are you seeing any hints, any signs? And then we'll conclude with your concluding remarks. But where do you see? What's your next step?
As I told Izadi, you caught me at a little sad time. I was very upbeat and optimistic, and I thought like educating people will make a difference. And I was really working hard in that direction. But looking at how things are turning out, people do not want to be educated. People want to prefer to turn a blind eye to data. But you can't argue with data. I think I will still continue to do this type of work with the hope that the newer generation, there are so many good people out there, and if this sort of education can make some difference, then I guess these are the people who will be in ODAC meeting in future.
Maybe we should put you in ODAC.
Then probably that will make a difference. But ultimately, like regulatory policy is or reimbursement policies, these are one part of the equation, but what matters is actually what happens in the clinic between the physician and the patient. That's the ultimate endpoint of all these policy decisions. So if through this work, a number of physicians will stay more updated about these issues, and they will try to make a rational decision making with the patient, then that is what ultimately matters. And as more and more physicians become aware, I think physicians and patients, we hold the ultimate power, right? If, like, let's say there is a trial being offered at our center, and the physician there is aware of all these issues, and the physician says, no, I'm not going to recruit any patients to this trial because you are using an inferior control arm, I will not accept that, or you should have a crossover design built in, then that's how I think change can happen. It will take time, but I think that's the ultimate end goal. I'd like to say one thing, like you know him very well, Professor Ian Davis from Monash University, Australia. He's one of my role models. I really admire him. He has led all these big prostate cancer trials. And when I published this paper, his comment, I can't quote exactly, but I'll paraphrase. His comment was, this is a good checklist that I will keep as a reference before I start any trial, or before I start writing about writing my trial publication.
I loved your article. I loved it. I'm like you. I just don't know if we're ever gonna live to the day. Well, let me back up, Bishal. I think the cynical person in me feels that there is never a perfect trial. I mean, you do peer review, you provide critical appraisal of articles and papers, and you do a great job at. I bet you any trial that I will show you, you can definitely find some shortcomings and some problems with it. In fact, that's why in every single paper, there's a section called limitations of this trial. You have to have that section. Otherwise, you're never going to get the paper published. So I don't know. There's no perfection, but we don't want to have perfect be enemy of the good.
Yeah, but there may not be perfect trials, but we can be nearly perfect consumers of research, right? We can interpret the research well, we can use it better, and implement it in a very thoughtful way when we are making those decisions. So we may not be perfect trialists, but we can be perfect consumers of clinical research.
Oh, you can't beat that as an ending for this episode. This is really wonderful. Before I let you go, I just want to, first of all, congratulations on this work. I want to have you more often. This is such an enjoyable thing. But I want you to have concluding remarks, and you have to say something in Nepalese, in Indian, and in Japanese for listeners. But concluding remarks.
Thank you, Tadi, for this opportunity to share my thoughts and this paper with a larger audience. And I had missed seeing you. This was a great opportunity to see you again, although virtually. I'm going to say in Nepali that नेपाललाई अझै धेरै सहयोगको खाँचो छ। कोरोना भाइरसको कारणले नेपाल र नेपाली जनताहरूले धेरै दुःख पाइरहेका छन्। हामीलाई आधारभूत कुराहरू जस्तै अक्सिजन, आइसियु बेड, यिनी आधारभूत कुराहरूको खाँचो छ, अझै जनचेतनाको खाँचो छ। मलाई आशा छ सम्पूर्ण विश्व समुदायले नेपाललाई पहिला जस्तै, हामीलाई भूकम्प आएको बेलामा जस्तै, अरू बेलामा जस्तै सहयोग गर्नेछ। नेपाल र नेपालीलाई तपाईहरुको खाँचोको जरुरत छ। धन्यवाद। So I just pleaded to the world community to help Nepal in our fight against coronavirus. So in Hindi, I'm not a very good speaker of Hindi language. I understand it very well, but when I speak, I might be making some mistakes, so I apologize beforehand. भारत को भी सहयोग के बहुत जरूरी है। आप लोग जो भी आप लोग सकते हैं, भारत को जरूर सहयोग कीजिए। भारत और भारतीय जनता इस वक्त कोरोना वायरस के कारण आपत में है। आपकी जरूरत की आशा है। So I pleaded again to help India and Indian population.
Yeah, and I've been really, I've been very vocal on social media. It is so important. I've been very disappointed, I'm not going to lie, with the US response. We're seeing some movement right now on social media. But I apologize for interrupting. Go ahead.
And finally, about Japan. 今年日本でオリンピック
I even know that in Japanese. I know Japanese. I know.
やってるから皆さんできるだけ応援してください。日本でもこういうコロナウイルスのせいで結構大変だと思ってますけど、でも日本と日本人はとても頑張っていつも前向きに頑張っていると思いますので、世界中の皆さん、日本のため、日本人のためと、それともオリンピックゲームのため、ぜひ応援してください。ありがとうございます。
And you were saying that you won the Olympics, or good luck with the Olympics, I think.
Yeah, well, what I was saying was basically Olympics is happening this year in Japan, and I know it's a very difficult time because of coronavirus, but knowing Japan and Japanese, they are very hardworking people, and they will do their best to make this successful, and we'll need the support of everyone throughout the world, encouragement from everyone throughout the world to make this a successful event.
Well, you are one of the most talented physicians I know. Not only you speak 4 or 5 languages and you do research, and you just amazing. I think I'm just very happy that you were able to take some time and be with us. And I hope a lot of people listen to this episode. I think there's so many pearls in terms of clinical trial design in this particular episode. Keep up the good work, Bishal. We need to have you more often on the show.
Thank you very much, Adi. It's a pleasure. I hope we can meet in person sometime soon now.
Very soon. Very soon.
Yeah. Okay, take care. Thank you.